So I'm very happy to introduce you to Lee from Accelerated Biosciences, who is supporting our meeting, together with Kurt Abdolmonsen from NIH. But please take the stage, give him a warm welcome.
Thanks, everybody. Happy to be here every year. This is the best place to be, really. My name is Utah Lee. I am the CEO of Accelerated Biosciences, and we're working with the human trophoblast stem cells, which we get from an ectopic pregnancy. And these are the earliest cells that you could possibly source without any ethical issues. And we provide scale and immune privilege. Now, last year, due to extenuating circumstances, my collaborator wasn't able to come up to stage to speak. But this year we're back. So I'm very happy to introduce you to Kurt Abdolmonsen. Come on up. Okay. Thank you.
Hi, everyone. Thank you so very much for having me at the RDD. Thank you, Lee, for your patience. Thanks for the organizers, Morten, Alex, and Andrea. And yeah, this year I was approved to travel and present our work at the RDD. Hopefully next year and the year after, everybody will be approved, of course, by the US government to make it to conferences. So today, as Yuta explained, we are working on the human trophoblast stem cells derived secretome, which includes exosome or extracellular vesicles. And we are trying to find the effect of the secretome or the extracellular vesicles on senescence and age-related diseases. So this work is done in collaboration between us at the NIA and several labs and Accelerated Bioscience. I'm sure you are all familiar with this slide, but I just want to say or emphasize that the aging process is not just a simple decline. It is driven mainly by active changes in cellular processes. And one of these mechanisms, of course, is cellular senescence, which is a major hallmark of aging. As we all know, senescence, as we learned from Dr. Campisi a long time ago, that senescence itself is antagonistically pleiotropic phenotype. What does that mean? It means like when we are young, senescence is beneficial. It can suppress tumorigenesis, it can promote wound healing, etc. But unfortunately, in older population or at older age, we see that senescence starts to speak a toxic language in terms of secreting pro-inflammatory cytokines, chemokines, which spread in the tissue and promote age-related diseases, including cancer, fibrosis, and many other diseases that are associated with senescence. So now we start to consider senescence as a pathological condition, not just a process that we're handling, not just phenotype, but it's also associated with many different age-related diseases, including fibrosis, degenerative diseases, cancer, and so many different other diseases, because it secretes those toxic material, including the SAF, senescence-associated secretory phenotype. It allows it promotes the development of age-related diseases. That's why we and several other labs are interested in developing approaches or strategies to target senescence and senescent cells. As we heard in the previous session, the process or the experimental work on parabiosis is quite beneficial for the old animals. So this is one nice strategy. This strategy proves that the secretome might actually be working very well. And many other speakers already talked about programming or reprogramming. Also the CAR T cell therapy, also targeted therapy. We're also developing and interested in senolytics and xenomorphics. Senolytics are actually killing or eliminating senescent cells, while xenomorphic are trying to deplete the bad behavior of senescent cells in terms of suppressing the senescence-associated secretory phenotype. And several other speakers already mentioned caloric restriction and dietary interventions that might actually reduce the burden of senescence. Since already in the previous sessions, it was mentioned about parabiosis. Since it benefits the older animals, then we thought to test also the secretome that is coming from stem cells. And generally speaking, the secretome from stem cells would include exosomes or extracellular vesicles, EVs, and non-EV particles. So we thought we asked this question, what happened to the senescence phenotype and senescence-associated traits if we treat cells with the secretome? This is just a quick overview about the exosomes. They are secreted by almost all cells, and they are generated in the endosome, secreted to the outside of the cells via multivesicular bodies and microvesicles. These extracellular vesicles, they are membrane-bound vesicles. They are full of bioactive molecules, including protein, mRNA, microRNAs, long non-coding RNAs, and other soluble proteins. But they also, on the surface, they are decorated by tetraspanins that are commonly associated with the exosomes or extracellular vesicles, including CD9, CD63, and CD81. These are considered to be secondary messenger, or messengers that come from one cell to be received by a different cell or recipient cells, and then they change gene expression profile in those cells. So before asking the question, what is the effect of the stem cell-derived secretome or exosome on the recipient cells, I personally thought too much about what kind of stem cell shall I use. And two major criteria that I really needed to address before any acquisition of secretome. Number one, it has to be ethically correct because of my personal interest also. I don't want to have any issues with the NIH or any issues with the ethics regarding the stem cell source. And the other issue was how early can I get the stem cells. And during my search, I found Accelerated Bioscience, this genius work on acquisition or obtaining or sourcing the human trophoblast stem cells from ectopic pregnancy. So that also saves the women life. If that pregnancy stayed in place, it will most likely kill or rupture the fallopian tube and leads to the mother's death. So as Yoda explained, these are highly pluripotent cells isolated from the ectopic pregnancy between 4 and 8 weeks post-fertilization. So these are very active cells and very early in stem cell lineages. So I collaborated with the Accelerated Bioscience to get the media, the secreted media from these cells. And then we decided to plan the study by testing the effect of these exosomes or extracellular vesicles on senescence, and also characterize the secretome and the EVs by themselves, and also test them in aged mice and age-related disease as well. So here what you see, our first study or first analysis, we obtained the secretome from the human trophoblast stem cells, and we also isolated the extracellular vesicles. We induced senescence in WI38 cells and using ionizing radiation, and we also tested other models of senescence, including doxorubicin, etoposide, and we observed similar results. So we treated the cells with ionizing radiation to induce senescence, and we also added the secretome. And also on the other side here, we added the exosomes or extracellular vesicles. And as you can see here, inducing senescence and adding the secretome at day 3 and harvesting the cells again on day 5, we observe at the mRNA level a significant reduction of mRNAs that encode for SAS proteins, including IL-8, GDF15, ICAM1, and BAF as well. The exosomes, or the EVs, also recapitulated that effect, so we could see a reduction in these mRNAs that encode for SAS factors. So this is mRNA, and this is like a small screen of a small number of the SAS encoding mRNAs. However, we also did the RNA sequencing to see what happened at the global levels after treatment with the secretome. And as you can see here on the volcano plot on the left side, there is a wide suppression of SAS-related mRNAs, indicating that the exosomes and or the human trophoblast stem cell-derived secretome also suppress the mRNAs, many more mRNAs, as you can see on the left side, including IL-6, IL-8, and CXL1, and so on. Since that wasn't the mRNA, we also wanted to know what happened to the secretion of those cells. What is going on in the secretion of those proteins that are encoded by these mRNAs. As you can see here, this is O-link proteomics, which revealed a significant downregulation of so many SAS-related mRNA, SAS-related proteins. And here on this side, you can see that they were all IL-6, IL-8, they were mostly downregulated in the secretion in senescent cells. And there are other proteins that were, of course, upregulated. We also did that at the individual cytokines. And as you can see here, these were measured by Bioplex, ELISA assay. IL-6, IL-8, GDF15, they will always being downregulated after the treatment with the human trophoblast stem cell-derived secretome. So, with a focus on the exosomes or the extracellular vesicles, to test them and to characterize them properly, we isolated them using the size exclusion chromatography. And as you can see here, we confirmed the isolation using the electron microscopy. And you can see here the membrane-bound vesicles and also the markers on the surface. When we isolate the exosome from certain cells, we relate them to the parental cells, where the parent cell, where they come from. And we know that the human trophoblast stem cells, they do express a good number or large number of longevity-related genes, and also they have high plasticity, and the proteins associated with these cells are often involved in or promoting tissue repair. How about the exosomes? We observe that these exosomes reflect large number of stemness, and it's pretty exciting, pretty interesting to see that the protein profile of these exosomes are actually reflecting broad stem cell signature of mesenchymal stem cells, any kind of, or adipose tissue stem cells, bone marrows, and even stem cell eye. So indicating that these cells can differentiate to anything, and they can also reflect large number of stemness, of stem cells. We also see that these cells are actively secreting pro-repair factors, and you can see here some of those proteins like pentraxin-3 and several collagens that are normally downregulated with age. We see that they are secreted by these cells in the exosomes. Importantly, we see in the pathway analysis and computational analysis, we see that the secretome itself is pro-repair, pro-growth, and activating different interesting or important signaling pathways in the recipient cells. This is very exciting for us because we normally know that senescent cells, on the blue side here, these are the proteins that are downregulated in senescence. And as we look at the proteins that are expressed or enriched in the exosomes, we see a good number of proteins, 130 proteins on, we can see here. These proteins are downregulated, normally downregulated in senescence. So it seems like the extracellular vesicles are bringing extra proteins that are otherwise downregulated in senescent cells. And these proteins are important for multiple pathways and multiple physiological effects on the cells, including mRNA stability, DNA repair, DNA replication, and even also the extracellular matrix. So we think that the exosomes or the extracellular vesicles coming from the stem cell drive, the human trophoblast stem cells, are restoring or potentially restoring some proteins that are otherwise missing or downregulated in senescent cells. That drove us, actually, this is actually the summary of what we're trying to say. We tried to find the mechanism, how these extracellular vesicles are actually suppressing the SASP phenotype. And as you can see here, as I mentioned earlier, there are some proteins that are involved in DNA damage repair, and we confirmed that the effect of these exosomes by doing the COMET assay and Western blot analysis. And this is the COMET assay showing us in cells that are not treated by the exosomes or extracellular vesicles, they showed large tail in the comet assay, while cells that are treated with the exosomes, they don't show much of DNA damage. And that you can see here down here with the exosomes and the conditioned media as well. With the EVs and the conditioned media, they both downregulate the phosphogamma H2X, which is a DNA damage marker. We also observe a less phosphorylation of NF-kappa B, the P65, which is a regulator of SASP transcription. It's a transcription factor that goes to the nucleus and start activating the transcription of SASP mRNAs. So in summary here, we observed that the exosomes or the extracellular vesicles coming from the human trophoblast stem cells, they do suppress SASP most likely by improving the DNA damage repair or improving or protecting cells against the DNA damage, and subsequently you can see less SASP production at the mRNA level and also at the secretion, at the level of secretion. Finally, I want to mention our pilot study that we're performing right now, and we're trying to expand the work on these animals. We started to inject the conditioned media or the secretome into aged mice, and we can see that for a month. And then after that month, we can see some significant reduction in age-related factors in the circulating system, including GDF15, CXL1, DPP4, and BAF. And these have strong indicators in age-related phenotypes, including cancer, hypertension, and we're actively pursuing these further on a larger cohort, not only at this level, but also at age-related diseases. And here I would just like to say I'm very happy to collaborate with anyone who has a model to explore age-related diseases. Please let us know. Yoda Lee can definitely, he has been so generous with us, and he provided the secretome, so he can also provide the cells. If you need exosomes in your age-related model, please let me know. Please let him know, when we can certainly collaborate to expand this work. Currently, what we're doing with the help of several PIs at the NIA, with Dr. DiCabo, we're expanding this as well. And with Isabel Bierman, we're expanding this to test the effect of this secretome and exosome on stem cell exhaustion. With Dr. Josephine Egan, we started to look into diabetes. And with Dr. Ranjan Sen at the NIA, we started to look at the immune activity. With that, I would like to thank everyone involved in this. Dr. Miriam Garospi, of course, for her support and the lab. Dr. Kapayanis in the clinical investigation branch, and of course, Dr. DiCabo. Wonderful work helping us in the translation gerontology. And of course, Dr. Lee and his company, Accelerated Bioscience, thank you for helping us accelerate the work. And of course, thank you, Dr. Farouji, for helping us with the fund to keep this work running. Thank you very much, everyone, and I'm happy to take any questions.
Thank you so much, Yuta. We have time for one or two questions.
Hi, very nice talk. So I think all of the models of senescence you guys looked at were DNA damage-induced senescence, right? And the SASP itself is quite heterogeneous depending on how senescence is induced. I'm wondering if you guys looked at oncogene-induced senescence or secondary senescence based on primary senescent cells inducing.
You're right. So far, we tested etoposide-induced senescence, doxorubicin-induced senescence, ionizing radiation. But we will definitely test other types of senescence, of course. Thank you. Sure.
All right. Thank you so much, Kat. That was really fantastic.